Okay.
Again, thank you for that kind introduction. And Katy and I are also co-chairs of the steering committee, and we're happy to represent the consortium, the Human Biomolecular Atlas program today. The talk is gonna start off highlighting some of the things that we've been developing as part of HuBMAP over the first 4 years of the project. And I'll cover kind of the basics of what HuBMAP's all about and highlight some of the things that we're doing kind of on the analytical side of things. And then I'll pass it over to Katy, and she'll take over talking about some of the wonderful tools that are being developed through the Hive, some of the bioinformatics groups and computer science groups, and then also highlight on a high level what the goals and aspirations of HuBMAP are during what we call the production phase during the next 4 years of the project. So to start off with, the vision of HuBMAP, really what we're trying to address is the comprehensive molecular mapping of the human body at cellular resolution. When we think about this within the consortium, we generally kind of break that down into 5 primary goals. The first of which has been a particular focus over the first 4 years of the project, is the development of the tools, the techniques, the protocols that enable next generation highly multiplexed molecular mapping at high spatial resolution. I think one of the things that makes HuBMAP so unique is, although most of the data we've generated in the first 4 years is still 2-dimensional in nature, a primary goal of HuBMAP is to generate our atlases in 3 dimensions, and that'll be the primary focus during the production phase of the project in the next 4 years. Of course, HuBMAP, like all consortium, is hyper-focused on establishing an open data platform that will allow us to integrate and visualize the data that each of the data producers are generating, but also, of course, rapidly make these data findable, searchable, interpretable by the greater research community. HuBMAP, again, like most consortium, is also kind of built from the ground up to support collaborations, both within the consortium, from day one, bringing together the analytical groups with the computational groups so that we can work together to build tools was a major thrust within the consortium, but also making sure that the tools that we're developing are being implemented and distributed to other consortia. So working hand in hand with other consortia like KPMP, like HPAP and other things, other consortia. So that's been a major focus. And then NIH and HuBMAP is also, particularly in the later phases of the project, going to focus on supporting pilot projects so that ultimately we can demonstrate the value of both the computational tools as well as the data and the atlases that are being produced. Now, the way the consortium is organized is really broken into 3 primary components. The first of which are the tissue mapping centers. Our group are one of the tissue mapping centers, and these groups are really focused on a broad range of aspects of the consortium, everything from tissue collection to developing the technologies and the assays for molecular mapping, generating data, and building molecular maps for our particular organ. We work very closely with the transformative technology and development projects and the rapid technology implementation projects. These groups are developing either supportive technologies to some of the things we're doing in the TMCs, or they're developing kind of new and up and coming technologies that once validated, and can operate at scale, then, can apply to become a tissue mapping center. We also, of course, work closely with the HuBMAP integration, visualization, engagement component. And so this is a critical aspect of HuBMAP, and I think a really a powerful resource of what's been established during the first 4 years is this amazing team of computational scientists that are taking the individual maps that we're generating in the tissue mapping centers and building true atlases. So connecting the data to ontologies, as you'll see later on in the talk, and building the tools that allow these data to not only be ingested with the proper metadata, but also then the tools to visualize the data and analyze the data so that it can be disseminated to the general public. So together, it's a pretty wide range of technical expertise that is available within the consortium. Everything from folks that are focused on single cell analyses of various types, multiplexed imaging approaches, whether that's protein assays, RNA assays, or, as you'll see some of our work later on, lipid and metabolite imaging using mass spectrometry, and then, of course, the necessary tools to bring all that together to assemble maps, to build atlases, and make connections across the spatial domain using things like common coordinate frameworks. Currently, HuBMAP is made up of over 350 researchers from 28 funded groups, and kind of the breakdown of those groups is highlighted here. So we currently have 4 funded RTI projects, 8 funded TTDs, 11 funded tissue mapping centers, and 5 funded Hive groups. And these groups really represent institutions from across the United States and around the world, as highlighted here on this map. So many of the states are represented, as well as research groups in Switzerland, Netherlands, and United Kingdom. So it really is, becoming even more so a worldwide effort to generate these molecular atlases. Our primary focus is normal human tissues, and we're working on a wide range of organs, which is a great opportunity to make connections between these organs, but it is a challenge in building tools that are flexible enough to be able to address many different biological systems. And so here's just a list of the organs that we're currently focused on. The initial set of organs, we got kind of a 2-year head start on those, and then more recently, those with the asterisks were added about 1.5 years ago. And so this is kind of our final list of organs that we're working on. Now, during the setup and scale up phase, which has really gone over the last 4 years up until, I guess we kind of draw the line in the sand, if you will, in September to now entering the production phase, we're really focused on developing the tools. So the analytical workflows, the standards for metadata and protocol tracking, so all of this data can be managed and reproducible. Generating some of our initial multimodal, multi-scale data sets, and then building up some of the tools that will enable the 3D mapping going forward. And you'll hear about a lot of these things, whether it's the ASCT plus B table or some of the work with Azimuth, building the reference data sets and the 3D maps. Creating our version 1 of the HuBMAP portal, which is available. I think Katy may have linked to that in the chat, and if not, I'll do that when I finish up talking. And then, of course, establishing some of the initial outreach and collaboration programs. So we are making connections with specific other consortia, but we've also established our own programs within HuBMAP. So whether that's summer internship programs or jumpstart programs for postdocs and early investigators, and you'll hear later on about some of the things we're doing to engage the broader research community through Kaggle competitions. Now, in addition to covering a wide range of organs, we're also uniquely, I think, covering a wide range of analytical technologies. So this is a schematic that we've put together to kind of highlight the broad range of capabilities that are being deployed throughout the various tissue mapping centers and other analytical sites within HuBMAP. And in this schematic, we're kind of placing all the technologies in context of molecular coverage and specificity and the kinds of molecules that they're targeted for, as well as spatial scale, from kind of whole organ analysis on the left through subcellular, single cell and subcellular resolution on the right. And so as you can see here, we have groups that are generating data using kind of the traditional in vivo imaging strategies. We have groups that are generating bulk analysis still to kind of support some of the data that's being generated in single cell and the spatial assays. So we have a whole suite of single cell technologies that are being employed, as well as spatial transcriptomics. As you'll see from our group and others within the consortium, there's a handful of us that are also working on high spatial resolution imaging mass spectrometry to target things like metabolites, lipids, and proteins. Of course, the fish-based assays, as well as the antibody-based imaging technologies, most of which are highly multiplexed or kind of three-dimensional in nature, light sheet and three-dimensional confocal, for instance. So again, a wide range of analytical capabilities, which is a big opportunity for integration of data across scales, both in terms of spatial scale and kind of molecular classes. But also it does introduce some challenges, and the Hive has done a really good job of building the tools necessary to bring all these different data types into the fold. And so this is it's a lot to take in. These are just kind of all pretty pictures to show examples of some of the beautiful data that are being generated within HuBMAP. So things like two-dimensional imaging mass spectrometry, three-dimensional imaging mass spectrometry, average anatomical atlases generated through in vivo imaging, light sheet, the various forms of highly multiplexed immunofluorescence and fish-based assays, and of course outputs from single cell analyses are all being generated and are all available on the HuBMAP portal currently. Now, I do want to take just a moment to highlight some of the things we're doing with our tissue mapping center, because I think it's a good example of the kind of multimodal nature of the data that we're collecting and how we're specifically developing our assays so that we can link to some of the tools that Katy will talk about through things like ASCT plus B tables, so we can make connections for even untargeted molecular imaging data specifically to cell types and functional tissue units. So our tissue mapping center at Vanderbilt is focused on kind of two primary workflows that are very tightly interconnected. First of which is what we call a multimodal imaging mass spec workflow. And it's going to combine imaging mass spectrometry with various forms of microscopy. So again, we can make connections to specific cell types and functional tissue units. And then on the other hand, spatially targeted multiomics, whether that's spatial proteomics or spatial transcriptomics. And the combination of these two workflows together really allow us to map across a wide range of molecular classes, from RNA to proteins to lipids to metabolites, and across wide spatial scales. As you'll see, we're getting pretty good on the spatial resolution, so we can certainly focus and get deep molecular profiles for kind of multicellular functional tissue units, but also we're approaching single cell resolution for most of these imaging technologies. So very briefly, imaging mass spectrometry is performed by taking thin tissue sections. We apply a multi-matrix. We then go from one pixel to another, firing a laser, generating ions that are made up of the endogenous molecules at that pixel location. We then perform a mass spec experiment at every pixel location. So you can think of each pixel as its own mass spec experiment. So we're detecting thousands of molecules within every pixel. So we have a mass spectrum for every pixel. Any of the peaks or molecules within that spectra we can represent as a heat map, or what we call an ion image. And this is what some of the data look like coming off of these systems. So we've made a lot of progress at our site in terms of improving spatial resolution so that we can again link molecular data to kind of the structures within the biological tissues to give us that level of biological specificity. So this is an example of an overlay of 6 lipids. These are false color scales, so they just represent, the colors represent intensity, which is a proxy for abundance for these lipids. And we're picking out different structures. We've just selected these because they're highlighting glomeruli within the tissue, proximal tubules, distal tubules. We're just selecting molecules that are associated with different structures within the medulla of the kidney, which is up here towards the top, as well as the cortex. And of course, these are all human tissues. So these are tissue blocks from nephrectomy samples. So just an example of kind of the multimodal nature. Again, this is the primary focus of what we're doing in HuBMAP, is bringing these things together. So this is an example of a couple of forms of microscopy that we can generate. We're also generating highly multiplexed immunofluorescence, but we can take these microscopy data sets and learn something from it. So we can bring in immunofluorescence, we can train models for segmentation. And once we have that, we can create segmentation maps, and that's what you're looking at here. So for tissues that we run with imaging mass spectrometry, which have deep molecular information, but they are untargeted, so we don't have kind of a knowledge base of what molecules should be in which structures. We can then overlay segmentation maps like I'm showing here on the right. So this is using the various forms of immunofluorescence as well as autofluorescence microscopy to segment the structures of the nephron and map that over these whole slide images. And then we can use computational approaches to correlate our imaging mass spec data with the various forms of microscopy. And so what you're looking at here are example lipids and metabolites that are highly correlated with the collecting ducts, the distal tubules, the glomeruli, and the thick ascending limbs. And again, the whole point of this is that we can link lipids and metabolites to the specific ontologies, in this case for the kidney. And you'll hear about some of those tools from Katy. And last thing, again, to kind of bring home this kind of multimodal nature of what we're trying to accomplish within HuBMAP, we're also, our group and others are using this kind of molecular and spatial data from the microscopy and the imaging mass spectrometry to then go collect things like spatial transcriptomics data, where we can now get deep molecular profiles from specific regions of the tissue. And so one quick example of this is shown here. So this is, again, human tissue, where we've segmented based on the microscopy and identified important features based on the imaging mass spectrometry. In this case, we're using the nanostring GeoMX system where we can target very specifically these functional tissue units and get transcriptional profiles. And so in this very simple example, we're just comparing the difference in the transcript profiles from the inner and the outer medulla for collecting ducts, for instance. So looking at specific differences in gene abundance, and then we can also look at pathway analysis. So for instance, one of the biggest differences between the inner and outer medulla is the TCA cycle, and these are the associated genes with the TCA cycle and highlighting the difference between those that are upregulated in the outer medulla versus those in the inner medulla. So that's a lot to kind of unpack in terms of some of the things we're working on. But the important thing is from a resource standpoint, not only are these technologies being developed, but we're working very hard within HuBMAP to publish and track protocols very specifically. So on protocols.io right now, we have 181 published protocols that are publicly available, highlighting all of the assays that I just highlighted, as well as those being developed by the broader range of tissue mapping centers. So it's an immense resource of protocols that have been validated for each of these assays and that are publicly available. And you can just search HuBMAP on protocols.io to find these. All right, Katy, I'm going to pass it off to Katy now, and she'll take it from here.
Hi, thank you. Good to be here. Really, really nice to see this effort you are all working on. I browsed the DK website to learn a lot about this effort. And I think there is a lot of interest also within HuBMAP, of course, to very systematically scale up the mapping of the human body so that we can collectively create a map of the human body at single cell resolution. And it really takes a village. And so you might wonder how you can possibly take data from so many different tissue mapping centers and other experts and bring it together in a common coordinate framework, and then analyze it using many different types of assays, and then ultimately zoom from the whole body down to the single cell level across anatomical structures into functional tissue units, which Jeff just showed, and again down to cell level and maybe even subcell levels. And so what you see here is the registration user interface that was developed to make it possible for different teams to indicate using human expertise where a tissue block came from. So here you have this yellow block that in the beginning is kind of on the side, and then you have to move it to the part where it came from. And we realize this is actually, we run user studies, so it's plus minus 1.3 millimeters that human beings can do that, depending also a little bit on the organ and how many structures you have that can be used as landmarks. But ultimately, this is very helpful for the second step of tissue registration, which uses the biomolecular information that is in these tissue blocks, but also in the reference atlas, to then do the stage 2 registration. So again, it's a two-stage process, one using human intelligence and then using machine intelligence combined together. On the left-hand side here, you see the anatomical structures, and actually just like in a video game, these anatomical structures collide with the tissue block as you move the tissue block in. And you can see this in the lower right-hand side here of the animation, where you have the anatomical structure tags that you can change as an expert, because you might like to have a few others, or you might say, well, in my tissue blocks, this anatomical structure does not exist. I just can't put it in a way that it really would do the right collision. You also get to see on the top right here that you then indicate the size of the tissue block, the number of slices if you plan to take tissue sections, and you also, of course, keep track of the rotation of the block. So you have ultimately the size, the location, and the rotation. And you really need to know where that tissue section and tissue block came from, because it matters. If you take it from the kidney upper cortex, it's very different if you take it from the hilum, and we just need to know this. Ultimately, this will help us build the atlas. So on the next slide, you see the exploration user interface. So after many teams have registered their tissue, currently we have 5,000 data sets registered from different consortia. So this is HuBMAP, GTEx, Spark, and some other consortia. Then you can explore what tissue exists across the male and female adult healthy body. And you can do this either by running a search for demographic metadata, such as sex and age and BMI, for instance, or by searching for anatomical structures here in the top left. You can also start to search for cell type, because many people want to really understand what cell types can be found where. Ideally, also what expressions of genes and proteins can be found there. So that's the next step deliverable. But then as you click on a certain tissue block, you will see on the right-hand side show up different data sets from different donors, and you can then explore them. On the next slide, you do see the spatial search. So here you have selected one tissue block, and actually now on the right-hand side, you have more information on who uploaded that data, how many tissue sections exist, what types of data exist for that tissue section, because oftentimes you can now do 2 assay types per tissue section, or you have multiple tissue sections and you run different assay types for consecutive sections. And then that data becomes more and more valuable also for atlas design, because we absolutely want to have a better understanding of what gene, protein, lipid, and metabolite biomarkers are most descriptive for certain cell types and states. And so now on the next slide, you get to see the new and enhanced spatial search interface. And the animation doesn't run right now, but that's okay. So what you can do here is to increase the size of a so-called probing sphere. It's like a golden sphere that you can increase in size until you cover more and more volume. And everything that's inside of that volume will be returned as a search result. You then get to see not only which anatomical structures are inside of that spatial search query, but also which cell types can be typically found in these anatomical structures as predicted from the anatomical structure cell types biomarker tables. And given that the animation doesn't run, you might like to go to the exploration user interface and just try it out yourself. It's actually fun to play with that data. So on the next slide, you get to see that if you now click on one of these data sets on the right-hand side, you can explore that data in much more detail. So here you go over to a data set from the right kidney, that's MALDI-IMS data, showcased in the Vitesse browser that Niels Gelenburg's team at Harvard Medical School has implemented. And you can then explore that data set in much more detail. You can zoom in, you can also download the data set now and get a lot more information. So on the next slide, you have the VR organ gallery. Those of you which happen to have an Oculus or would like to have this as a Christmas present, please do contact Andreas Bückler. He's always looking for beta testers that can provide feedback on this evolving set of organs, which can now be explored in that 3D space. But you can also now explode all the organs out of the body and then explore all the tissue blocks that have been registered into these organs in a very 3D manner, and including you actually get to see the physical size of these tissue blocks. And if you are interested in learning more about it, let me know or contact Andreas at abückler@iu.edu. On the next slide, you have more information on the ASCT+B tables that Jeff and I have been mentioning. And this is an invention by the Kidney Precision Medicine team. They started to publish one of those tables in one of their papers, and we believe it's a wonderful way to bridge across scales. So you have an anatomical structure partonomy on the left here, where you start with the human body and then major organs which are inside of that body. And inside of each organ, you have anatomical structures, functional tissue unit, down to the single cell level. For each cell, you have not just the cell name, but you also have the crosswalk over to the cell ontology, just like you crosswalk the anatomical structures over to Uberon or FMA. And then you have a bimodal network from the cell types over to the biomarkers. And the biomarkers might be genes or proteins or lipids or metabolites. We also have proteoforms. And with that architecture, you can now start to keep track of what biomarkers are commonly used to characterize certain cell types, which cell types are typically located in what anatomical structures and how these anatomical structures build up to the whole body ultimately. And so using that infrastructure, you can then also link over to ontologies, again here to the Uberon FMA ontology and to the CL ontology. And you can also start to connect this agreement on the naming of cell types and biomarkers and anatomical structures to physical representations. So for the 3D organ library objects, we use the Visible Human Project data to represent coarse anatomical structures that help subject matter domain experts register tissue at that plus minus 1.3 millimeter range. And then Jeff just showed his work, his team's work on identifying functional tissue units in kidney. And those can now also be brought in to provide physical manifestations of the typical size and location and density of certain anatomical structures within the context of the kidney cortex, for instance. And so you have the naming, just like in a normal cartographic map, you have to agree on names. So this is coming also from ontologies, and we realize that we have to extensively extend these ontologies to actually cover the healthy human body. But you also have the 3D, and in some cases still 2D, physical size and shape of these 3D references, which are then ultimately representing the anatomical structures and cell types. So in the next slide, you see the combination of these 3D structures here, a spleen in 3D. And of course, you just see the outer shell, but there's more internal structure. And then you also see the white pulp of spleen as a 2D illustration. And you can refer them back to the anatomical structures that are captured in the anatomical structures, cell types, and biomarker tables. And you also see these different linkages that now interlink the anatomical structures to cell types via located in relationships, and between biomarkers and cell types as so-called characterized relationships. On the next slide, you see that it's really not just KPMP and HuBMAP and GTEx and many others funded by NIH that are working on this together, but also different consortia that are supported by the Human Cell Atlas and that are enjoying European and other funding. And on the right-hand side, you see the evolving list of organs, and you also see which consortia are caring about what organs. So some are squarely focused on just brain, and that's complicated enough, right? Others are squarely focused on kidney, but then there are some efforts such as GTEx or HuBMAP, for instance, which are taking on quite a few organs, and we are in the need of creating scaffolds and infrastructures that really extend to more than one organ. And that's really a wonderful visualization challenge, but also a socio-technical challenge, right? How do you interlink so many different types of data sets? How do you harmonize them? How do you link them to existing ontologies? But also, how do you do the socio-technical part of it, and the social part of it, so that technical experts and engineers and tissue experts and single cell experts and pathologists and surgeons and anatomists all can talk together. So on the next slide, you see the azimuth reference that you might have seen before. So Rahul Zahid at the New York Genomics Center, he is working on expanding substantially as a number of references that can be used to do cell annotations for new data sets. So using these azimuth references, you can take a new single cell RNA-seq data set, upload them to the azimuth hubmapconsortium.org website, and then get automatically assigned cell type names. And that really improves the rigor by which these cell type names are assigned, but also scales it up enormously, because in former times this was a very manual, very time-consuming process. And so he has a huge community buy-in to this, and you might have seen that there are 3 new references which just recently became available for heart, adipose, and tonsils. On the next slide, you have the portal, and you might like to check out the portal so that you see not only the tools presented here today, but also some of the other tools that our tools components have been implementing, including also a sales API and of course the Vitesse browser, which you see here. So on the next slide, you see the evolving set of HuBMAP data sets, and also color-coded here by the different types of organs. And the number of organs is steadily increasing. I think we now have 28 ASCT+B tables, and quite a number of more assay types than listed here. So we have to update, this slide. On the next slide, you have the visible human MOOC. For those of you which would like to learn more about how the data comes into existence in HuBMAP, or which like to have an ontology 101 session with Mark Musin, or would like to know how the exploration user interface now also exists in the GTEx portal, you're very welcome to come into this MOOC. It's an open and free course which anyone can take. And in fact, we oftentimes use it to onboard new experts to the HuBMAP project and other projects that aim to map the human body at single cell resolution. And there are also some rather fun sessions and modules in here, and a lot of self-tests and training tests. So feel free to check out these materials. On the next slide, you have information on HuBMAP training. So we had really great undergrads come in this summer actually, and we will have an expanded program next summer. So if you have bright undergrad students, you should definitely point them to this opportunity. And this is also going on since 2 years now, and you see all the undergrads listed which attended in 2021. So as you see, there's a growing number of undergraduates which come into technical areas, but also into the tissue mapping center wet bench labs. So it's a quite diverse experience that these different students enjoy. And then of course they also help cross-pollinize and kind of transfer knowledge from one lab to the next. And we also had 3 Jumpstart awardees in 2021 on the lower right here. And it was really wonderful to see what these young investigators were able to do with rather small amounts of money, but they had a big bang impact. And it's just great to see them rise through the ranks here. On the next slide, there is an upcoming event at the Cornell University campus at Roosevelt Island in New York City. The event is scheduled for March 1st to 3rd in 2023. Everybody is welcome, and there is a website with more information forthcoming. If you're interested to learn more, just send us an email. I don't have an email on here on this slide, but it's going to be epic. So please come. I think we have a number of really cool speakers, and we would love to have as many junior investigators as we can fit into the room during a pandemic. On the next slide, there's more information on the Kaggle competitions. So one just ended. I put the link into the chat. You might like to check it out. So the plan is to have more and more algorithms developed, not just within HuBMAP, but by inviting the larger Kaggle community and experts there, potentially using algorithms developed in very, very different domains of research. And here we were interested to segment out different functional tissue units across 5 organs. And the teams did very well. The performance prize winners already enjoyed their competition prizes. The scientific prize winners, we are still deliberating. The judges are still meeting on that one, but they will be announced very soon as well. So if you are interested in Kaggle competitions and would like to participate in future competitions, we have 2 more planned on the vasculature CCF, the Common Coordinate Framework that uses the vasculature system to get us a latitude-longitude system for the human body. So the link which you see down here would get you to a paper that explains a lot more about the vasculature common coordinate framework. And we believe this actually has a good chance of succeeding. And therefore, we are putting up 2 more Kaggle competitions in that realm. On the next slide, So Jeff mentioned that much of what we are presenting here is coming from the pilot phase. The production phase just started. We are all super excited that we are now scaling up and trying to run some of the analysis and visualizations at scale, which is non-trivial. We are very, very interested to take on 3D maps and reference data sets during the production phase. In fact, many of the human reference atlas ambitions cannot be delivered on if we don't have 3D data. Like, for instance, having a vasculature system as a coordinate framework absolutely requires that you can follow blood vessels, and that's really, really hard if you just have tissue sections. We also will need to agree on the common data types that we want to acquire for every single tissue blocks that is used in the atlas generation process. We need to build bridges between different data types. So Rahul's team is taking the lead on that, and we are also very interested to work with other teams on generalization of cell by gene matrix data. We now have azimuth maps for quite a number of organs, and you also have listed here those experts which have spent many, many hours of their time, oftentimes also their free time, to help create some of the maps, but also, of course, these azimuth maps. These are cell gene matrices which plug right into the ASCT+B tables for the cell types and the gene biomarkers. But then, of course, there are also protein biomarkers. That's the last item on the list here, where we have validated antibody panels and validation reports. And yes, all of those have IDs, and they then are used to construct so-called organ mapping antibody panels, or OMAPs. And again, that's the cell types by protein biomarkers matrices that are also in alignment with the ASCT+B tables. We have started to identify and generalize, I think that's on the previous slide, the functional tissue units in 3D, and we are very excited to collaborate on those. Now on this slide here, we have more information on demos and collaborative projects. So as you see, we are very interested to integrate with the matrisome database to identify organotypic vasculature, to reverse engineer ovarian organization, to identify mitochondria variants, and also to work on multi-tissue analysis from single cell donors, to basically take one organ and get tissue from that one organ shared across different tissue mapping centers, and then compare the results of different assay types. And also very important and near and dear to all of our hearts is outreach and collaboration. So we will continue these junior investigator meetings, the jump starts and summer internships and Kaggle competitions. But we are also organizing joint workshops and meetings. There's a plan for enhancing diverse perspectives that every single team in HuBMAP had to promise to fulfill. And we, first of all, had to construct it in alignment with what is already in existence at our universities, but also what can be done together across institutions. And we have a number of open working groups. I checked out who is on the call here, and I see many familiar faces, and it's wonderful that many of you are already part of the affinity reagents, the data visualization working group, which is a new one, but also the ASCT+B working groups, which meets every first Wednesday of the month, and has really been instrumental in helping us coordinate efforts across so many consortia. I think that's the end of the slide deck. We would be happy to answer questions. Thank you very much for inviting us, and looking forward to see what questions are in the chat, or feel free to speak up also. Thank you.
Yeah, thank you, Katie and Jeff. So now is our question and answer time. You could unmute yourself, or you could raise your hand and then type your question in the chat. So let's see, I think during Jeff's presentation, there was, I think Anita asked a question and Katie already answered. Is antibody all have or the antibody have RID? And then if you haven't seen the answer in the chat, you could see, yeah.
There's a follow-up. So it looks like, thank you, Katie, for pointing me to your documentation. it looks like RIDs have been required as a part of your schema for some time. This is, by the way, this is Anita Vandrowski, and I run the RID initiative. So one of the things that we're trying to do is to gather data about validated uses of antibodies. And so I was trying to poke around at the site to see if I can, find any antibodies that have been used and to pull the data that way. So is there a way for that kind of a query to happen? I wasn't seeing it on the front end.
Jeff, would you know?
Yeah, I can. Yeah, I think, that is something we've been talking about, is the ability to kind of query based on whether it's an antibody or a lipid or a molecule. Basically, query on the molecular side is probably, I don't want to speak for the Hive groups, but I think, that's one of the things that's been brought up as kind of a next step for the portal. So it is something we're talking about, and I anticipate that will be possible. And as far as RIDs, yes, it's a required aspect of what we do in generating the antibody validation reports and ultimately creating the OMAPs. So it's certainly a priority for us.
Did any of those make it out into the literature?
So there's always a delay, right? In these things. So if I can speak for our tissue mapping center, for instance, that will be coming in the next 6 months or so. So we're working on finalizing our AVRs right now, and then those data sets are going to be up shortly.
And then for the HuBMAPs, Andrea Radke at NIH, she has been very proactive moving this effort along. And I think there are a number of papers which are either under review or have been accepted but not yet published. So I think there are a number of papers coming out, but everything that's out right now is on the HuBMAP's website. And I have put that link into the chat. I can do that one more time. But great question. I like these questions.
Okay, so how about how can others help construct or use the human reference atlas? Like if a research wanna contribute or wanna help to build the reference atlas, how do they do they need to join as a member or just contact or through the website?
That's a great question. So we are currently reviewing all the ASCT+B tables that have been submitted for the Christmas release of the Human Reference Atlas, the fourth release of this atlas. And we are actively looking for experts on the 28 organs that we are now covering. So if you go to the CCF portal and then to the ASCT+B tables, you would see a listing of those. So if you have expertise for a certain organ, you might actually be curious to see these tables, and there are very detailed instructions on how to best review the anatomical structures patognomy, to review these cell types. So you might have your own data set of cell by gene matrices or cell by protein matrices, or you maybe have looked at lipids or metabolites for a specific organ. We would very much like to hear from you and would then share with you an official invite to review the table for your respective organ. And of course, it's not just the table, it's not just the naming, it's also the 3D and 2D spatiality of the anatomical structures and functional tissue units, which are functional tissue units still in 2D as illustrations. Because ultimately we would like to make sure that we don't just have comments and authors from one school of thought, but that we are really getting a consensus organ where scholars from around the world can really weigh in and help us all understand what is known so far. And in other cases, it's just now that the data becomes available. And so, as Jeff pointed out, the Hive, and I'm representing the technical part of the Hive here, is in the process now to take the atlas, to take the Azimuth reference systems and the OMAPs, and to connect them with data that is available in HuBMAP, and that might have come into existence before we had those references, but also new data that just now is uploaded to the portal, to connect these two, but then also to see what other data exists that's high quality and that could or should be connected to the Human Reference Atlas. And then we have not only the provenance from textbooks and from scholarly papers, but also have provenance information on what data is used and has been used to construct the atlas, providing more and more evidence for the accuracy of the anatomical structure, cell types, biomarkers ultimately.
Thank you, Katie. There is one question from chat. From Marian, how does the ASCT plus B tables interact with FMA and Uber?
Excellent question again. So the ASCT plus B tables are changed by subject matter domain experts, organ experts, on a continuous basis. And then 2 times a year, they are frozen, they are reviewed, they are DOI'd and published, right? These are the release cycles we have. But every Monday, the tables are read out by David Uzumi Sutherland's team at EBI in the UK, and they are compared with HuBMAP. And HuBMAP is an ontology that consumes and contains the human healthy part of HuBMAP. It also has now more and more elements of FMA which are missing in HuBMAP, especially from the vasculature side. So FMA, the foundational model of anatomy, captures much more information than HuBMAP currently does for vasculature. So we are ingesting that into HuBMAP and into HuBMAP. But HuBMAP also has the cell ontology. So every Monday we compare the ASCT plus B tables with the HuBMAP, which represents the existing ontologies, and then create a so-called validation report. And that validation report is instrumental for us to understand what tickets have to be added for including new anatomical structures and cell types into the existing ontologies. We believe the existing, or maybe I should say I believe, the existing ontologies might only cover 30% of what is needed to really represent the healthy human being. And this is a guess right now because we just don't know. We don't have the data yet. But all I have seen in the many, many meetings we had so far, it's not 90% that is in there. It's much, much less. And so interested in advancing these existing ontologies, we have to find ways to get so many new term requests issued, and then also made a substantial and integral part of these existing ontologies. On the biomarker side, I think we are better off. The existing ontologies have a much better coverage. But in terms of the microanatomical structures that researchers seem to care about, and the cell types and cell states that seem to matter to understand healthy human function of cells, we just don't have the sufficient numbers. So if you just require your researchers to use HuBMAP and FMA or cell ontology to tag the data they have, they will not be able to do it because these two existing ontologies are not expressive enough, or they're just not complete enough to do that right now.
Okay, I think Mary write in the chat about the HuBMAP is truly breathtaking. And then so are there any question in the chat? You can type in or unmute. There is a question, it's listed under the key question that HuBMAP can answer. Maybe already mentioned in the presentation, but how about just do a summarize or just let audience know what assay types are best to map the human body in 3D and across scale.
I don't know that it's one assay type. I think, I think what we're finding is that each assay has its own, obviously has its own advantages and disadvantages. So I think it's really a multimodal strategy and an integrated strategy that's critical. So things that span the breadth from single cell and spatial transcriptomics all the way down to the ability to map metabolites and lipids. It's really critical, and that's a challenge because for certain assays like light sheet microscopy, for instance, you can map a few targets in 3 dimensions, but to get kind of broad scale deep molecular coverage in 3D takes new technologies, and those are the things that are being developed. And so those are the things I think we're most excited about. I know Katy is excited to work with the 3D data, and some of the things that our group and the other tissue mapping centers are developing are going to kind of usher in some really unique capabilities in terms of 3D multimodal imaging.
And maybe to add to this, it's actually a feature, not a bug, is that we need to map at multiple levels. So you could hope for something where you can just have a lot of single cell data and then just puzzle it all together because you have so much data. But it turns out that it's still quite expensive to acquire high quality single cell data for even smaller tissue blocks. And so we cannot have we do not have the luxury to take, let's say, 10,000 carefully selected human beings across different ages, different sexes, different ethnicities, different BMIs. We would love to then sample very systematically across the major organs, but we just can't do this. This is what you would do if you would do a cartographic map of the world, right? You would send out your surveyors and you would do this in an extremely systematic way. But of course, there you only have one Earth. And you only have to do it one. You don't need the 10,000 or more specimen to have a better understanding of what differs and what is shared across human individuals. So what we can do, however, is to map at different levels. So there's different types of data, which we have a lot in the healthcare system. And that's more at the gross anatomical level. We have a lot of liquids that are regularly drawn from human be it blood or urine or other parts, and that's not necessarily currently used in HuBMAP, but it's used in other projects, and I think it's very, very valuable for understanding health. And then, of course, now you go down to the more high resolution data sets, which is the super exciting part. And that's where a lot of high stake papers also are currently published. And now you get to the data, which is currently quite expensive, but gives us new views of our world, which is really magical. And yet now you can just zoom into this level and map at these different levels. So you might have seen that the azimuth references also exist at different levels. So for the kidney, for instance, you have a more general level and then level 2 and a third level. And this is by design, because some data sets might only be mappable at the more general level, whereas others much benefit from having the highest resolution cell type annotations. And similarly, also for the exploration user interface, it is at the coarse anatomical level, but then you can zoom in using VTES to the single cell level. And so I think just like we need to have user interfaces that help you explore the human body from the gross anatomical scale to the maybe even subcellular level, you will also have to find a way to map new data into this multi-level atlas.
Thank you. And then I think we have probably just a few minutes left, and then just quick to summarize the last question, like what common coordinate system CCF is best to construct the human reference atlas?
Jeff, do you want to do it first answer and I do a second?
Sure. Yeah, I think what we're finding is that similar to the assay question is that we probably need more than one framework. And, you know, so you can rely on Cartesian coordinate systems like we are right now, but that, you know, I think we're most interested in kind of more biological driven coordination systems, whether it's through vasculature or the nervous system. You know, we can use different landmarks to relate our data to one another. You know, I think during our more recent annual meeting, the analogy for atlas building is, you know, you can have maps that relate things to location relative to rivers or roads, or, you know, there's different ways to kind of relate these data sets together. And so, and it's similar to the assay side, I think there's opportunities for using multiple CCFs together.
Yeah, I agree. So ultimately, there might have to be combinations. So for some organs like the brain, you have the skull, and that has been used quite successfully to create a coordinate system for the brain. In other organs, you have very distinct biomarkers that can be used as landmarks. So you actually have not anatomical structures or bone structures, but instead you have biomolecular structures that help you find out where you are in a larger organ. And in some other cases, you might need a combination of all three of them. But that's quite different still from having a latitude-longitude coordinate system. So here, you would be looking at a way to have an address for every single cell in the human body. And that's, again, different from taking a cell type population and trying to map it into a human body. So those of you which are interested in kind of latitude-longitude information systems and address systems, you might like to go back to that one slide, which talks a little bit more about the vasculature common coordinate framework. And I will also put that link one more time into the chat here. I have to just go get it. So great question again.